physiology. And the main thing we'll be talking about is Spermatogenesis. Now this process takes place in the seminiferous tubules, and it's about 70 days long. It's basically the formation of sperm. Formation of sperm in the seminiferous tubule. Process takes roughly 70 days. And that's the process. Mitosis, meiosis, spermatogenesis. Mitosis is cell division. You already know that. And it's the regular cell division where the daughter cells have the regular amount of diploid DNA. Now in repro, when the germline cells undergo mitosis, as long as mitosis happens, a man is fertile because those cells are dividing. You always have sperm. So I'm writing a man will remain fertile into different books and they say different things. 60s, 70s, you have read, because they always have mitosis. And think about what a woman does with mitosis. It stops before she's born, when the follicles form, right? And there's menopause in women. There's no menopause in men, really. But there's a defined menopause where there's depletion of eggs and ovulation stop. That happens about age 50.5. Could happen in your 40s, could happen somewhere in your 50s for a female. But men, mitosis happens throughout their life. So that's one gender difference, one sex difference. So meiosis is the same meiosis we've been talking about. 1, 2. Meiosis 1, meiosis 2. We'll go through it again. Because the goal is to become haploid. Spermatogenesis after you finish dividing the DNA, spermatogenesis is the cell undergoes morphological changes to become swim ready. Cell becomes swim ready. That's a much larger distance to go to fertilize the egg. Here's a picture of a. Well, there's a lot going on here. We kind of draw it out here. Here's a whole seminiferous tubule with a lumen in the middle. And imagine a compartment that's more outer and more inner. Okay? There are these columnar cells, but you usually can't see them. There's a cell called the Sertoli Cell, and let's say this is it, or tentacular cell. And there are these tight junctions between them. Okay, I draw a couple there. Tight junction, tight junction. The tight junction is a junction between cells that is tight, but it does allow things through it. Now, you usually don't see these cells as a whole because they're covered by the developing sperm. But what's important is the tight junctions between the sustentacular cells create this outer compartment, say from here to here, called the basal compartment. And then we got the lumen here where the sperm will swim away eventually. But then you have this compartment next to the lumen, adjacent to it, called the adluminal compartment. Okay, so this whole thing is the seminiferous tubule where spermatogenesis is happening. So spermatogenesis is again these three steps. And it's occurring pretty much all in here. In the seminiferous tubule. They pretty much happen in this order too. Meiosis, Spermatogonia just undergo meiosis. You can kind of start to look at the figure here. In the basal compartment, the Spermatogonia just undergoes regular cell division where it's diploid and then it divides and the two daughter cells are diploid. Okay, call them type A, type B. So it's like you got a cell, it divides, becomes two cells. That cell's a spermatogonium. And it divides, you get two cells, one's an A and one's a B. So that's mitosis occurring in the basal compartment. The reason why they designate 1A and 1B is because you want some of those, half of them, to stay in the basal compartment and to continue to undergo mitosis, so you'll always have sperm. You want the other half to actually to become sperm. Okay, so that's why they give the A and the B designation. So for mitosis, they say that the type A remains in the basal compartment as a stem cell. It'll undergo mitosis, more mitotic divisions. So type A remains in basal compartment. Basically will continue mitosis. B, it kind of slips past the tight junctions and is pushed to the adluminal compartment, and it will begin meiosis, meiosis 1. So I'm writing push to adluminal compartment. Undergoes meiosis. So I'm going to try to sketch this out here. Erase this for now. So what you see here, the yellow is the cytoplasm of a sustentacular cell that I said you can't see because the cells that are becoming sperm, cover it. And so if you can kind of draw that out here, this might take me a little while. Just drawing like the cytoplasm that'll surround all the sperm that are surrounded by them. Let's see here. So what I traced in purple there is just, there's a nucleus of a sustentacular cell. I just traced all the yellow stuff that surround the cells. And what you got there is, I'll draw the cells a different color. Like for example, I'll give you a second to draw that if you're drawing it. All right, that first cell I drew is the spermatogonium undergoing mitosis, cell number 1. In this part of the lecture, keep track of the cell name. Don't call them sperm now, because it means nothing if you're trying to study. If you call them all sperm, it's meaningless, and you won't get any question right on the exam. Spermatogonium is what we're going with here. And I said they undergo 2 types of, when it divides, you have 2 cells. You know, A and the B. I already defined them, A, B. So I'll just put them under spermatogonium. It's the B that are pushed and enter meiosis. So that cell, you can call it a primary spermatocyte, the ones that enter meiosis. So let's put a 2 next to the B, assuming it's pushed the adluminal compartment, and call it 1 primary spermatocyte. So you go from spermatogonium, primary spermatocyte, and I remind you, this is meiosis, the same meiosis. At the start, you got the 23 tetrads. It's diploid replicated. 23 homologous chromosomes that are replicated, and we call them tetrads. Now, meiosis 1 occurs, prophase 1, metaphase 1, etc., and you finish, and you get 2 cells that are called secondary spermatocytes. Get 2 cells in here. Let's call number 3. So I have 2 secondary spermatocytes. It's the same terminology as primary oocyte meiosis 1, secondary oocyte meiosis 2, same thing, right? This is meiosis 1 for primary spermatocyte. You finish meiosis 1, you enter meiosis 2, and each of those 2 cells are secondary spermatocytes. So at the start, in meiosis 2, each of these cells is going to have its diploid, not diploid, it's haploid replicated 23 dyads. Yes, that's the correct ploidy and R 23 dyads. You do meiosis 2, all the steps, and now you're going to have 4 cells. They're called spermatids. I guess go with early spermatids. Well, spermatid is the term, early and late. Forget early and late. Okay, you finished meiosis 2. Now you got 4 cells. 4 spermatids. And each of those cells, you should know this. What's the ploidy? What am I going to put? I'm going to put NR? No. What am I going to put?
N.
Just N. Yeah. N is haploid. Remember what N means. N is a set of homologous chromosomes. Just one set. So remember, your cells don't have one set. Your cells have 2 sets. Right? The other set will come from the egg. So, but this is what you want for reproduction. One set, 23 singles. 23 single chromosomes. So spermatogenesis is 3 processes. Mitosis, that's pretty much number 1. Pretty much what we just did was meiosis 1 and meiosis 2. And meiosis is the other step of spermatogenesis. The last step is to become swim ready, and that's a process called spermatogenesis. Not shown here. I mean, you got the terms here too. You go meiosis, then you go spermatogenesis. Basically, you go from regular looking round cell to a rocket, a missile-shaped cell. The process of becoming swim ready. There's no more moving around the chromosomes. You're done with meiosis. It's just morphological cell changes. So that's spermatogenesis. The process of becoming swim ready. And then when you finish, you're ready to break off the tubular wall. That's called spermatozoa. We draw some cells here. Just draw a little rocket ship in there. With the head stuck in the sand. Spermatozoa. I've been saying sperm this whole unit, but now I'll use the right term since I've made it to this lecture. Spermatozoa is the full name for that cell. Sometimes people say spermatocyte, spermatozoa, same thing. So the process of becoming a regular round cell to a rocket-shaped cell that's swim ready is spermiogenesis. The whole process from top to bottom, about 70 days. Maybe 16 to 18 days at number 1, maybe 24 days around here, number 2. Number 3, the secondary spermatocytes, never see them. As soon as you hit meiosis 2, you start. I mean, so these are very short-lived. Maybe they hang around for a day, and maybe this is another 24 days. The point is, the whole thing is 70 days. The last 24 days is the spermiogenesis, where you become sperm ready. Let's take a look at that. What you should notice from this is, well, we're not going to look at the details of it. I just show it to you because I want you to see that the more immature cells are in the basal compartment. When you get closer to the lumen, that's when you start to see the rocket-shaped cells. They're about ready to break off and swim away. Okay, so that last process where you become swim ready is illustrated here. And when you have a regular round spermatid, 24 days, the last 24 days of a 70-day process, boom, now you're swim ready. The details to notice are that the Golgi, let me zoom in a little bit. The green Golgi, see that now. The green Golgi is making a helmet over the purple nucleus. And that, the Golgi becomes a structure called the acrosomal cap, okay, or the acrosome. So in a completed sperm there, you can see the acrosome forming a helmet over the nuke. And yeah, the structure here. So let's kind of draw that out just a little bit. Oh, before I erase this, are there any questions? I'm going to talk about some details of spermiogenesis that are in here. Okay. So I guess what's could be difficult is getting a handle on the term spermatogonia, spermatocyte, whether it's primary or secondary spermatid, you know, the processes, spermatogenesis, spermiogenesis. They all mean something different. Okay, but they all sound very close. Spermiogenesis. Notice the cell loses most of its cytoplasm. And we said that the Golgi becomes what's called the acrosome or acrosomal cap. Cap around the nuke. So say you got nuke. And haploid DNA in there. It's got a double membrane acrosomal cap around it. And the acrosomal cap, the Golgi's got all these vesicles with lytic enzymes. So, not to go too fast, purple is nucleus. It's got the goods, the genetic material. That's utmost importance, the genetic material. But the acrosome is important. I'm writing, it contains vesicles with lytic enzymes that will breach the egg, breach the ovum, bust it through, okay? Bust the wall, literally. Contains lytic chemicals. That punch holes. As we'll see, when you get to the egg, there's these walls. Like this is a wall. One is called, it's a wall of cells called the corona radiata. The other one's the zona pellucida. We'll note that when we get there. Anyways, for now, got the acrosome, and around that, put a plasma membrane, cell membrane. So on the warhead there, yeah, like a little mid-piece. And tail. Before I finish drawing it, we have real pictures of this in my slides somewhere. Here's a blueprint of it, of the different things I want you to know. Head, neck, mid-piece, tail. So the neck is kind of like, it allows the head, pretty much here to here, to pivot, okay? The mid-piece, it's got a lot of mitochondria in it. Let me finish drawing this. Here's I'm going to draw a lot of red circles in it to illustrate that the mid-piece contains lots of mitochondria. You need energy for whipping the tail for locomotion. Head, neck, mid-piece, tail. That's the basic idea of your swim-ready cell. Well, if our drawing's not to your liking, just look at the real picture. It's much better. So you're going 17,000x, which I think is remarkable that you can do that. 17,000. I said you got to go to 400 to see cells, and that's our wimpy slides, our wimpy scopes. They have 17,000, and you can see the center axis, you can see all the MI is mitochondria. PM, what do you think that stands for? Plasma membrane. What do you think AC stands for? Acrosomal cap, yeah. It's 14,000x. And so, yeah, it's a beautiful looking cell by itself. That's one little sperm. And that's what you need to know about the spermatozoa, the basic parts that will get us through this lecture. Let's move on and talk about the cells that nurse it, the sustentacular cell. I already mentioned them a little bit. Those are the cells that form the tight junction. What I didn't say is that tight junction forms a blood-testis barrier that protects the developing sperm. So I'm going to list this. Well, actually, maybe I'll leave it up there for a while. Those are those columnar cells in the seminiferous tubule. An older name that I still use is called the Sertoli cell. Sustentacular is, that's what you'll see in your book. Formerly Sertoli. You may see that on your test, so I throw both at you there. And they have a nurse function. Remember, their cytoplasm surrounds the cells. That's why I drew that whole purple thing. They're are surrounding it. Cells don't do good by themselves, they die. And you know, I know I erased it, but you notice how I connected all the cells together? Because they need that. They need to be connected. See this? See how they're connected? Right there. This is not an individual cell, it's kind of connected to this one. So I tried to draw that earlier. And, but these cells have sustentacular cells lose function. I already noted the tight junctions. Tight junctions between these cells create the basal and luminal compartments. And also form the blood testes barrier. Because, I'll underline that for importance, your antibodies or immune globulins would see the developing sperm as non-self and attack it. So it's a nice little barrier to protect the developing sperm. Okay, that's another reason for it. So I pretty much said everything that's on here now, paraphrased it. And one thing about the tight junctions is about the testosterone levels. Testosterone levels have to be high enough. Sometimes I abbreviate testosterone with the T, circle it. Testosterone, one thing, it helps the spermatogenesis process. In a young man, maybe in 20s, per gram of testicular tissue, the spermatogenesis can produce something like 6.5 million sperm per day. It's a lot. Spermatozoa daily. As you get older, you get less testosterone. And, 50s, 60s, maybe it gets down to 3.8 mil. Still quite productive as you age. But if testosterone gets too low for whatever reason, these junctions get loose. Low T loosens, I'll put quotes around it, that's kind of mean. They get less, they lose their integrity. And they allow the spermatogonia to slip through too fast, okay? And they never develop because they get through the tubule too fast. And maybe they get to the lumen and they're not swim ready and they just get absorbed there. They never make it. So I'm writing let's developing sperm. So low T loosens tight junctions. As a result, let's developing sperm through too fast and they never develop. So another thing that's worth mentioning is, once you're developed, there's a process of getting your head out of the sand and breaking off the tubular wall and swimming away. That process is called spermiation. It's not like there's not enough terms already. Anyways, it's my job to present them all to you. So spermiation, the process of breaking off the tubular wall. You see it there at the bottom, right? Head in the sand, I'm ready to go. Let's break off, boom, and swim away to the epididymis. Somewhere. The process of breaking off tubular wall. If you have low testosterone, but you know, you developed and you're ready to go, but there's low testosterone, you have trouble doing this. Can't break off. Can't break free. And what happens is you just stay there, and then eventually just the Sertoli cell just gobbles you up. You just get absorbed there. Let's move on. So testosterone, Sertoli cells, you know, it's all important. Here's some models in the room, and it's part of your lab study guide. And they're numbered 5, 6, and 7. But let's observe what we can see here. This is a primary spermatocyte, and it's undergoing anaphase 1. And if you look at each of these, it's a double. They're separating the homologous chromosomes. If this is anaphase 1, what is this? Anaphase 2. I see 2 cells, and because it's anaphase 2, these must be secondary spermatocytes. Now you're pulling apart the dyads. Each of these is a single. And it's hard to tell from the picture, that's why we have lab time. We can look at it up close and personal. And by the time we're finished, these look like spermatozoa to me. In each of these cells, what's the ploidy? And yeah, haploid, excellent. Okay. Here's the female counterpart models, 8, 9, and 10. This is, I'll just tell you it's anaphase 1. Okay. What's forming here? What would you call that? I said identify. It's a polar body, first or second? First, yeah. Let's go to the next one. This is anaphase 1. This is anaphase 2. Identify. First polar body, that's dividing. The second polar body is here. And this is anaphase 2, and pull these apart there. And here you've completed. Assume that sperm has arrived, and you complete meiosis 2. But all our polar bodies here, which one is this, first or second? Second. These 2 are from the first. Okay, and these are haploid chromosomes that would go with the sperm that would be here. But not shown. Sometimes what I like to do is kind of mix and match. Can you tell which one is the sperm? This one is. And how do you tell? The polar body, the unequal division of the cytoplasm here tells you it's oocyte female. The equal division of cytoplasm tells you it's male. Okay? So I'll ask you that, is this male or female? I don't know why I like that question. I ask it all the time. All right. Well, anyways, both of these are the same phase of cell division. Which is it? Anaphase 1. And both of these are the same. Anaphase 2. All right, so the hormonal control of spermatogenesis shown here is kind of like when I taught this for the female, but it's much more simple for the male because I don't have to teach a cycle. It's been like 45 minutes for the female. This will take me like 5 minutes. Let's clear the board. So what I see at the top there is our gonadotropin releasing hormone. It goes through the portal system, it targets the gonadotropes, and they release our two gonadotropins, FSH and LH. Which stand for follicle-stimulating hormone, luteinizing hormone. In the female, FSH developed follicles. Are there follicles in men? Yes or no? But we keep the name. And so I can memorize the second hormone name. Luteinizing hormone. Is there a luteal phase in men? No. But we just keep the name. But they target, the male thing. So now in males, FSH will help stimulate the Sertoli cells. This is tentacular cells. And they'll secrete, it says there, it says APB. ABP, which stands for androgen binding hormones. Basically molecules that will sop up the testosterone. ABP stands for androgen binding proteins that help bind up testosterone. The LH, those are going to stimulate, outside the seminiferous tubule, as you well know, as you identify them, they stimulate the interstitial cells of Leydig, or just interstitial cells, and they secrete the testosterone. Interstitial, let's spell that right. Secrete testosterone. So in testosterone levels, they help do a lot of things, make a male a male. Today we're concerned with, they help facilitate spermatogenesis. That's what testosterone is really good for. Facilitate spermatogenesis. That's the whole process, right? The 70-day mitosis, meiosis, the whole thing. So basically, when you have high testosterone, you increase the sperm count. That's a negative feedback. Yeah, these are the dotted line. So, low T, low testosterone, decreased sperm count. That would remove the negative feedback. That would be like a positive feedback. That would be like a stimulants. So this high-low thing, there's no cycle. It's just, we don't keep track. There's no cycle in men. But I kind of gave you the numbers. The daily output, 6.5 million daily for a young man per gram of tissue. Now listen to sports talk radio. They always have commercials for men. They always have these andro commercials, these supplements you take that increase your testosterone. And don't take them. If you take testosterone and you don't need these, what happens to them? They shrink. Okay, they just go. It's like you're just taking it. Why? Just let your body do it. Okay, unless for some reason you're under doctor supervision and they've identified a clear reason for you to take it. Okay, don't just. You should only take these things under doctor supervision, is my opinion. Okay. All right. Any questions on the gonadotropins in males? What? That's it. Just know what this does for that does for that, and that's it. In one of the early editions of Maria, she wrote that it's way more complicated in females. She actually wrote that. It's true. There's no cycle to teach, so it's straightforward. Let's talk about the physiology of the human sexual response. And no one even really looked at it scientifically. Masters and Johnson, usually their work is what is referenced to in most physiology textbooks these days. And what they kind of noted was the level of excitement, plateau, orgasm, resolution in the human sex response. So we'll kind of look at it in terms of the physiology here. We'll do males and females. For males, we talked about erection of the penis. It's a parasympathetic event, and I would note nitric oxide. That's probably the thing for male erection to note. Because nitric oxide is a vasodilator. It allows the corpus cavernosum to become engorged with blood when this happens as a result. What you're doing is you're increasing cyclic GMP, that's the second messenger, that allows the vasodilation to occur. There's also scrotal swelling, and their graph that I cut and pasted from their site has the level of excitement on the y-axis, time is the x-axis here. You can see the excitement here. So nitric oxide, the little blue pills, we know it now. Here's kind of a data graph from Louis Ignaro's early experiments. He's the one actually won a Nobel Prize for his work in nitric oxide. But nitric oxide vasodilates all blood vessels, not just in the genital area. And nitric oxide is levels are low, it's a huge risk factor for heart disease. Heart disease is your arteries getting clogged. When I studied heart disease in grad student, we always talked about nitric oxide. In terms of repro, he looked at, this is the muscle of corpus cavernosum. That's the erectile body that gets hard in erection. And this is from rabbit. And they've always known that nitroglycerin, NTG, had that effect, but no one knew what molecule actually did it. So he showed that nitric oxide is the molecule of nitroglycerin that has this effect, or you can have the relaxation, and that will help you achieve an erection, because when you relax, the tissue fills with blood. Okay, so whenever you identify a molecule and you can manipulate the cell physiology to it, and you can put it in a pill that you can ingest, I mean, that's how you can help people, right? Of course, that was done in the 80s. And when did we first start seeing the commercial for Viagra and Levitra and Cialis? Maybe early 2000s? It takes a while. In the plateau phase for the male, the penis is fully erect, the testes are all engorged. You may see fluid at the tip of the penis. That's the secretion of the bulbourethral gland. Let's remember that the function is to eliminate trace urine in the spongy urethra. Okay, so the orgasmic platform is a sympathetic response. So stimulation is parasympathetic, ejaculation is sympathetic. And so let's remember that I define ejaculation as movement of sperm out of the urethra with orgasm. So I'm writing ejaculation, movement of sperm out of urethra. Just to remind you of that. That's really what you need to know. And this kind of a, well, there's some details here that they note on the figure. Remember, sperm is released from the tail of the epididymis. You have contractions of everything here. The vas deferens contracts. The prostate contracts. The seminal vesicle contracts. There's a sphincter at the base of the bladder that contracts. And that helps to keep urine in the bladder during ejaculation, which is what you want. Okay. Do you remember the name of the muscle I said that provides the expulsive force of ejaculation? It wasn't listed there. The bulbal spongiosus. I'll list it there if you forgot that. The penis undergoes about 3 to 5 strong contractions followed by some weaker ones. They're about 8.8 seconds apart, expelling the semen. Okay, there's also a spike in cardiovascular. In males, orgasm is necessary for fertilization because the ejaculate has the sperm. Necessary for fertilization. For resolution, everything basically just goes back to baseline. And in men, there's a refractory period that's typically shorter in younger men. A refractory period is a period of insensitivity where re-stimulation is difficult or impossible. Okay. So in females, that same progression, you can see that Masters and Johnson noted different 3 general patterns of excitement and resolution. They're not all the same. But they all go through the excitement plateau orgasm platform. In excitement, there's increased luminal vaginal lubrication. Things swell. Basically, what they show here is that the cervix kind of gets out of the way. It's retracted, and the fornix kind of expands. Those are kind of the anatomical changes. So our remote uterus pulls up and fornix expands during excitement. That's kind of what's illustrated there. There's some lubrication by, we studied the gland next to the vestibular bulb, the greater vestibular gland. Okay. In the plateau phase, there's some color changes. The clitoris is retracted, and whatever max changes you observe happen here. Orgasm in females is similar to males. There's 3, 4, maybe up to 12 contractions of the orgasmic platform about 0.8 seconds apart. May be repeated. They may follow different patterns as shown there, the A, B, and C patterns. In females, orgasm is not necessary for fertilization. I mean, what is day 14, right? Ovulation. Well, that has nothing to do with this. Orgasm not necessary for fertilization. In humans. I've read that orgasm can trigger ovulation in other mammals, but not humans. It's day 14. But I've also read that if you have ovulated, orgasm can increase the chance of fertilization because oxytocin makes the uterus contract and it helps wash the sperm up to the egg for fertilization. All right. In resolution, everything relaxes, but there is no refractory period for females. So that is one different. I mean, Masters and Johnson's report, they can be multi-orgasmic up to 5 to 20 orgasms. So I would say women have all the fun. It's not even age dependent. So that's another difference. Men have refractory period, women do not. So refractory period here. No refractory period. Now that concludes those slides. Let's take a break now, and when we come back, I want to start development. Talk about fertilization.